Okay, so let's talk meiosis. So basics. The purpose of meiosis is for a cell to go from diploid to haploid, or a set of chromosomes, and it contains 2 cell divisions. And the meiosis is specific for germ cells or gametes and gonads. And germ cells and gametes are synonymous, according to my professor. Okay, so let's look here. So this is the whole process. Okay. Now, if we look at a diploid cell, we can see here that this contains 46 chromosomes and everything is normal. Now, when we want to enter meiosis, these chromosomes, they will be duplicated. So you will have 46 chromosomes and 92 chromatids. Now, after S phase and G2 and whatever, then you will enter prophase 1. And there are 2 important things in prophase 1. The first thing is chromosome condensation, and the second thing is homologous recombination. So those are the 2 main steps of prophase 1. And now if we want to get into more detail, so this homologous recombination, it has like 5 different steps. And so the first thing is, homologous recombination is not 5 different steps, but the prophase 1 will have these 5 different steps. So you have the leptothena stage, which just says that the chromosomes will condense. In zygothena stage, the synaptonomial complex will start to form, and it will hold the synapses between the 2 homologous chromosomes. In Pachythena stage, you can see that the synaptonomial complex is completed, and therefore crossing over occurs. So here is where HR will take place. And in Diplothena stage, you will see. Also, you will start to see the bivalent thing. Yes, and the synaptonomial complex will disappear. Will start to disappear in Diplothena stage, and now you can see the chiasmata. And the chiasmata are really important detail, because in anaphase, when the homologous chromosomes will start to separate, the only thing that holds these 2 homologous chromosomes, as you can see here, the only thing that holds them together is the chiasmata. And the chiasmata is formed when homologous recombination happens. So if homologous recombination doesn't happen, then there won't be accurate disjunction of the homologous chromosomes, and that's bad. Okay. So this I will not say everything that is included here, but I would like for you to read it, because it's the basics of crossing over and basics of homologous recombination, and so these are 2 different flashcards, but they're important, so read them. Okay, in case you would like to stop the video. Okay, so the next stage is the metaphase 1. Okay, and what are the purposes of metaphase 1? Well, there are 3. Okay, so nuclear membrane disappears. That's the first thing. And then secondly, the spindle formation. And thirdly is that the bivalent chromosomes will start to align on the equatorial plane. Align on equatorial plane. And as you can tell, now there might come up a question on colloquium where they will show you a picture of maybe meiosis 1, metaphase 1, and meiosis 2, metaphase 2. And they will ask you what type of meiosis this is, or if it's meiosis 1 or meiosis 2. I don't know, but like, anyway, the most important detail here is to recognize that in metaphase 2, there are no homologous chromosomes. And here you can see that there are 2 homologous chromosomes right here, and here you have only 1. And so if you get just a picture of this, and you get a picture of this metaphase 1, and they ask you to explain if it's mitosis or if it's metaphase or whatever it is, or if it's meiosis, well, then you can tell here that it's, well, first of all, it's a bivalent chromosome, and so that means that it has to be in meiosis. And because there is no homologous chromosome right here, well, then that means that it's meiosis 2. But anyway, so they align, and as I told you earlier about chiasmata, you can see that actually right here, it is only the chiasmata that is holding them together. Okay, now we go to anaphase. What are the purposes of anaphase? Well, it is, first of all, disjunction. So basically separation of the homologous chromosomes, and then also sort the homologous chromosomes into random combinations into the 2 different daughter cells. And that's it. And then telophase will happen, and the main purpose of telophase is just to separate the daughter cells, so you get 2 haploid sets of 2 haploid sets of chromosomes in 2 different daughter cells. That's that. And then you have cytokinesis, which is this part right here. So cytokinesis is kind of the same like in mitosis, so I won't go into detail about that. But what's important here is to know that, okay, so the daughter cells, they will divide. Well, actually here, 2 haploid sets of chromosomes in different poles of the cell. And then here you get 2 daughter cells in cytokinesis. But in cytokinesis, the cell will enter interkinesis, which is the interphase between meiosis 1 and meiosis 2. And it's important to know that interphase is the longest phase in a cell's lifetime. In a somatic cell, let's say, or whatever, because interphase will be like all this time, and then you will have the M phase, and that's it. But the interphase now is between meiosis 1 and 2, and it's really short. Like this one could maybe be, I don't know, from hours to lifetime for some cells, but in meiosis it's really short, because after meiosis 1, meiosis 2 will happen. Okay, now the main difference, so now when we enter prophase 2, meiosis 2 is exactly the same as mitosis. If you know mitosis, then you know meiosis 2. The only difference that you have to keep in mind is how they start. Prophase 2 actually starts with 23 chromosomes and 46 chromatids. And prophase 1, or not prophase 1, but just prophase starts with 46 chromosomes and 92 chromatids. So really that's the only difference between meiosis 1 and mitosis. Otherwise, this whole process is exactly the same as mitosis. No difference, kind of. But at the end you will get 23 chromosomes and 23 chromatids, and you will get 4 haploid cells. And that is meiosis.